3 and 4. Okay? So, and I know a lot of you had some questions about what's going to be on the exam. And pretty much anything in the class lectures in units 3 and 4 is fair game. And there's not really much more we can kind of narrow it down for you. Just focus on key concepts. Focus on specifics in so much as it seemed like a fundamental thing to know. And specific examples that really illustrate those key concepts. All right, so if I introduce a concept, basic idea, and then say, for example, if we look at such and such a system. Okay, so that's all fair game. Like on the first exam, I don't expect you to know every last little detail. There may be a few details in there, but if you miss a few, minor points, it's not going to kill you. Get those key concepts down. Okay? I know that doesn't. I know it's not what you want to hear, but that's what it is. All right. Yes. How many points is it?
Like 75.
It's going to be it's another 75-point exam, but it's worth a little bit more than the last exam. Your first exam was going to be weighted 30%. This one's going to be weighted 35. The final's going to be weighted 35. Just because there is more material. And the final is going to be cumulative. So the final will contain units 5 and 6, plus some material from the first 4 units as well. So you have another shot. If you kind of missed some of that membrane potential stuff, you can have another shot at it. So go back to your first exam and see if you can get that. Okay, so today we're going to talk about the chemical senses. And first we're going to talk about olfaction, then we're going to talk a little bit about taste. And then in the sort of the second half of today, and maybe a little bit into Thursday, we're going to sort of sum up a little bit of what we know about sensory coding. So we're going to talk about coding. Okay, so olfaction. Olfaction is a little bit of a different sense from what we've been talking about. And this is because it depends on being able to detect chemicals. Chemicals are physical entities and come in enormous variety. And there's really no way to sort of organize chemicals into sort of a single linear dimension, the way you can with sound, where you have wavelength is the sound frequency, or light, where wavelength is the color, and so on. Okay, and so olfaction is really sort of this weird thing. That it's a multi-dimensional sense. And as a result, you need large numbers of receptors. And in that sense, it kind of resembles immunology, where you have to be able to detect many potential partners for the receptors. And one of the hallmarks of the olfactory system, nevertheless, is that you have maps. We talked a little bit about sensory maps in other systems. And in olfaction, you also have some sensory maps, but only up to a certain level. And then we're going to talk a little bit about how timing is involved in coding, which we really haven't talked about yet. And we'll sort of wrap this all up in the second half of the lecture about neural coding. But we're going to sort of introduce the idea that timing of activity can be a really important component in the neural coding. Okay, we're going to talk a little bit about sparse codes a little bit later. So that's where we're going. And the sort of the end concept here is that what we're going to illustrate as a sparse code is really quite useful for learning large amounts of information in an associative way. So we're not really going to talk much about olfactory behavior, but you can think that as olfactory information gets associated with other information. And we'll talk more about this much later when we talk about memory systems. So, okay. So the sensory, what you're trying to sense in olfaction is the soup of chemicals that are in the environment that come in all sorts of shapes and sizes and contains different chemical moieties and so on. And so how are you going to organize this system? There really isn't sort of a single linear dimension, as we'll see. And furthermore, it's not just sort of single chemicals You're not just analyzing the scene to determine which single chemical. You're really looking for blends, right? So a floral scent of a rose is a blend of multiple chemicals. The scent of a coffee is a complex blend of chemicals. And when we sort of sense what we call an olfactory object, is really the combined, bound together blend of different chemicals. Okay, so there's kind of this tension between using it as an analytical sense, detecting specific components, and using it as sort of a synthetic sense, where you bind together different components into a particular percept. Okay, just to kind of give you an idea that these are some of the chemical structures. Of course, these are not just sort of flat two-dimensional things. These are three-dimensional objects, right? Molecules are three-dimensional objects. There's an extra dimension here that's not really represented on this kind of thing. But basically, you have very small chemicals and very large complex chemicals that are all detected, and they often are associated with particular percepts. Obviously, ethanols were associated with an alcoholic smell, and so on. And some of these are a little more salient than others. So the smell of urine or the smell of rotting meat have very particular chemical signatures. And furthermore, the chirality of molecules. So you can detect the differences between molecules that are identical, except that they're mirror image enantiomers of each other. So this is an example, a really well-known example of carbone that comes in two different enantiomers, and they really are perceived as completely different odors. Okay, so you're sensitive to that level of chemical structure. But of course, you're not aware of chemical structure. You're just detecting it as a sensation. Okay, so how do we start? So olfaction starts in the nose, obviously. You're detecting, when we talk about olfaction, we're really talking about airborne chemicals. If you're an aquatic animal, you'll be talking about aqueous chemicals, but we're not going to talk about that. So olfaction, for our purposes, is an airborne molecule that you breathe into the nasal cavity. Inside the nasal cavity, you have a sensory epithelium called the olfactory epithelium. And embedded within the olfactory epithelium are the olfactory receptor neurons. I'll often abbreviate these ORN. Okay, so olfactory receptors in the sense of neurons. Okay, and these are embedded in this epithelial lining, but sort of sticking out of that are the cilia. We'll look at those in the next picture. These olfactory receptor neurons actually have axons. So unlike photoreceptors or hair cells that make synapses onto afferents, they actually have their own axons that go through this bony plate and project to the olfactory bulb. So that's the first sensory relay. Okay, so you have these airborne molecules that come into your nasal passages, and they come into the mucus, and within the mucus, so here's an individual olfactory receptor neuron that sends these little protrusions out into the olfactory mucus. These are called cilia. Okay, and this is where the detection takes place. Here's the olfactory receptor neuron, here's the axons that are going on. And one interesting thing is that the olfactory system is known as one of the few places where you can get regeneration. So these olfactory receptor neurons are being regenerated every few weeks. So here's sort of a little picture of a dividing stem cell. And the theory is that part of it is because they're exposed to the environment. This is probably the most exposed neurons that you have. The retina, of course, is closed in your eyeball. So this is really quite exposed to the environment, and that probably has something to do with the fact that they need to be rejuvenated over time. And it actually it's kind of interesting because then it lends to a whole developmental question of, well, now that you have neurons, these neurons have to find their projection targets over and over and over again. We don't really have time to get into that. All right, so the cilia, we know, are responsible for the transduction of the odorants. Okay, we know that because if you apply odorants to the cilia, you can get a very nice, robust response. This is a voltage clamped current response. Remember, inward current is depolarizing. We're recording from the main cell body. Okay, but if you take those odorants and you apply it to the cell body instead, you get very little response. So it's quite clear that the cilia are responsible for the transduction of odorant information. And so here in the cilia, you actually have what's called a receptor potential. And then in the cell body, you have this kind of potential. This is called a generator current. So you have a generator potential. And then only when it gets to the axon do you have an action potential. Okay, so we can record these generator currents in a number of different neurons, and we can apply different odorants to the cilia and look at their response properties. So this is an example of one neuron that responds very strongly to cineol and isoamyl acetate and acetophenone. So it's sort of not very discriminating, but many of these neurons, neuron 2 and neuron 3, respond only to one or a very small selection of odorants. So they can be very, very selective. But in general, they can also respond to multiple responses. So it's not just single odorants, but they all have their own particular response properties. And you can notice that these are also tend to be very slow. Olfaction is a very slow sense, unlike the auditory system that we looked at before, which had very, very fast responses. Olfactory system is generally very slow. Okay, so if instead we record from the axons, we can record regular overshooting action potentials from these axons. And again, we can apply odorants. But now we're applying an odorant at different concentrations. So here's a very low concentration, you get a few spikes. Increase the concentration, you get a few more spikes. And increase it further, and you can get a larger number of spikes. So there's really appears to be pretty clear rate code where you have increasing concentrations of odors, you get increasing numbers of action potentials. It's pretty straightforward. Okay, and the transduction mechanism, another variation on the theme. We've looked at this in photoreceptors. We've looked at this in hair cells. So you might, when you're reviewing, might be able to want to compare and contrast different transduction mechanisms in the different sensory systems, for example. But this is particular for olfactory receptor neurons. So there's a number of different components that are familiar, but a little specific to the olfactory system. So first of all, kind of unusually, there's actually what's not pictured in the book very much, an odorant binding protein. So there are these proteins that are sitting in the mucus that are there to transport odorants. Oftentimes odorants, they wouldn't travel very well themselves in the mucus layer. So they're little proteins that will kind of help them along. And they'll deliver the odorant molecules to a receptor protein. So this is a very specific G protein coupled receptor. Okay, and we'll talk more about that in the next slide. So it's a G protein coupled receptor. When the G protein is activated, it's a particular G protein called GOLF. Okay, and it's very specific to the olfactory system. GOLF, the alpha component, will activate an adenylyl cyclase. Okay, and that'll create cyclic AMP. The cyclic AMP activates a cyclic nucleotide gated channel. It's a cation channel. Okay, and that is going to pass sodium and calcium, mostly calcium in this case. Okay, and that's going to depolarize the cell, and that's the basis of that the generator potential, the generator current. Okay, there's a couple of extra components here that we don't really need to get into. There's a chloride channel that's a little unusual that amplifies the signal. Okay, it's depolarizing, which is a little unusual, and that's because the chloride concentrations are a little different. It's very low in the extracellular medium, so chloride actually travels outward, and that's a depolarizing current. So a little amplification. And then finally, there's an exchanger, the sodium-calcium exchanger. Most of this current is carried by calcium, so you need a way to extrude that back out of the system. All right. So let's talk a little bit about the odorant receptor. So odorant receptor is in the protein that binds the odor. So olfactory receptor neuron, odorant receptor. Okay. And so this is, as we said, it's part of, there's this huge family of G protein linked receptor proteins. So originally, people knew, it was actually kind of hard to find this particular receptor. People knew that it depended on the G protein, but people looked for a long time and couldn't quite find it, and finally used the fact that it was probably a 7-transmembrane domain receptor, and used that to clone the olfactory receptors. Okay, and it turns out that part of the reason that it was hard to find was that these are expressed in very low levels because it's a very large family of various protein receptors. There are somewhere along the lines of 1,000 distinct genes for these odorant receptor proteins, and they all have variations. So there's variable amino acids and conserved amino acids, and that's because you have to detect all those different types of chemicals. So the system has basically taken this transmembrane, 7-transmembrane domain receptor, duplicated it, mutated it, and came up with this enormous family of genes. And it turns out that this accounts for about 3 to 5% of all of our genes. Okay, so it's kind of a crazy amount of genetic material dedicated to olfaction in the system. And you can compare that to the number of opsin genes. You have the 3 cone opsins and then the rhodopsin found in the rods. Okay. And this was discovered by Linda Buck and Richard Axel, and they got the 2004 Nobel Prize for discovering these odorant receptor proteins. Okay, so it turns out that each olfactory receptor neuron expresses probably only one of those odorant receptor proteins. And that accounts for most of the specificity of the olfactory receptor neurons to different odorants. Okay, and there's a whole line of evidence along those lines. Okay, so there's a sort of dogma of one odorant receptor to one odorant receptor neuron. And the olfactory receptor neurons themselves are only somewhat organized in the olfactory epithelium. So they're scattered over the epithelium in basically 4 rough zones. So let's look at that. So this is an experiment in which immunohistochemistry was done. So this is antibodies to a particular olfactory receptor protein, looking at the distribution. So if you remember from Unit 1, we talked a little bit about methodologies in order to localize particular proteins in particular areas. This is a good example of that. Okay. Okay. So the green here is GFP-linked antibodies stained for a particular olfactory marker. Sorry, this is a general marker for all olfactory receptors. These are specific markers for one or another olfactory receptor protein. Okay, so this shows you the whole olfactory epithelium, and then this shows you the distribution, each of these little green dots, the distribution of a particular odorant receptor protein. Okay, so it's found in these little punctates. Each of these dots is an olfactory receptor neuron in particular zones within. So this olfactory receptor protein is sort of in this area. This olfactory receptor protein is in this area. But you'll have to sort of imagine that they're also interspersed with lots of olfactory receptor neurons that are expressing other odorant receptor proteins. Okay, so you have this very heterogeneous expression. Okay. But kind of remarkably, all of those olfactory receptor neurons get together in the olfactory bulb. Okay, so every olfactory receptor neuron projects to the olfactory bulb and they project to a particular neuropil structure called the glomerulus. So here's the axons coming up and all converging on a particular glomerulus. Okay. In the glomerulus, the synaptic terminals of the olfactory receptor neuron axons contact the dendrites of the principal cells of the olfactory bulb, which are the mitral cells, also the tufted cells, two different flavors of principal cells, and those project onto their cortical targets. Okay. There are two types of interneurons in the olfactory bulb. We're not going to talk too much about this. There's the periglomerular cells. These are literally the interneurons that basically maintain contact in the glomerulus or across glomeruli and might be involved in sort of lateral inhibition. The granule cells sit close to the mitral cells and make lateral connections between mitral cells. So slightly different functions and different layers. Okay, but I really want to talk to you about this glomerular structure. In another slide. Just one small aside. We're not going to talk about this too much. So what we're talking about here is known as the main olfactory bulb. This is the main olfactory epithelium and the main olfactory bulb. But there's sort of a parallel system that's involved in specialized olfaction, the pheromonal system. And this is all we're going to really talk about it in this system. There's a particular area called the vomonasal organ in the nasal epithelium that has olfactory receptor neurons that express particular receptors for things that are of particular behavioral relevance to the animal. So in that sense, it's sort of a highly specialized system for very important behaviors, whereas the main olfactory bulb is more of a generalist sensory system. Okay, and the reason that you want this is that it's important for social behavior. So it's in order to detect conspecifics. It's important for maternal behavior. It's important for mating behavior. So these sorts of pheromonal signaling systems are quite important for lots of animals, especially rats. Rats and mice have very well-developed systems. And these project to a somewhat separate system, the accessory olfactory bulb, which then has higher order projections. Okay. And it's basically the sort of system is basically absent from humans. Okay. Our olfactory system in general is kind of degenerate. It's not nearly as sensitive and useful as a dog's system, for example, and almost any other animal. And so if somebody sends you an email trying to sell you human pheromones, don't buy it. It's a scam. Okay, so we basically have a pretty good general system, but not so much a pheromone system. Yeah.
So when we talk about like reduced olfactory activity in humans relative to like dogs and other stuff, is that due to diversity of receptors, or is it like a signal amplification thing, or like how does it happen?
Yeah, if I remember correctly, we do have fewer receptor genes. It's like they've been inactivated or lost along the way. And I think partly because we're so dominated by vision that we don't pay attention so much to the senses, the sensation that we do have. There are some people who are extremely sensitive, and people who are extremely well trained who can detect, notes in wine. People who, are involved in perfumery. They can like do that analytical pinpointing of very particular chemicals. So it's not that we're not really capable of it. We're less capable than a dog, say, but we also just don't use it very much. And we're so dominated by vision. Yeah. All right. So back to the main olfactory bulb. So this is a cross section through an olfactory bulb. And you can see a number of the layers sort of around the outside here are the glomeruli, that neuropil that receives input from the olfactory receptor neurons. And then you have a couple of layers here. The main outer layer here is the mitral cell layer, so that's the main cell bodies for the mitral cells. And then all of this coloration on the inside here is a granule cell layer. So there's an enormous number of granule cells in this system, and we still don't really know what they do. They're probably involved in some of the timing aspects. They're inhibitory. They may be involved in lateral interactions, but still sort of a really active area of inquiry. Okay, but it's very, very stereotyped that each of these glomeruli receive input from a particular set of olfactory receptor neurons that are expressing a particular odorant receptor. Okay, so that one-to-one odorant receptor to olfactory receptor neuron, and we showed how those olfactory receptor neurons are kind of scattered across the epithelium. They all converge onto a particular glomerulus, and in a very stereotyped way. So you can see this in animal after animal. And there's one on either side. You have like a left and a right side. So you have a glomerulus on the left and a glomerulus on the right. Okay, this is kind of a cool experiment. This is where you have LAC-Z staining, so it's sort of blue reactive dye in those olfactory neurons, receptor neurons. And so these are all the axons kind of bundling up and converging on a particular glomerulus. So you have this really kind of intense convergence of all of those olfactory receptor neurons. And so in a sense, it looks like, well, you know, our job is done. You know, olfactory receptor protein detects particular chemicals, goes to glomerulus, boom, you're kind of done, right? It's not quite that simple, because if you look at the activity tells a slightly different story. Okay, so here, this is, so these are data, this is from glomeruli in the Drosophila. So insects have a very similar system. I'll describe that in a little bit. This is an experiment of, I think, a mouse olfactory bulb. Olfactory bulb. This is using calcium imaging. This is using, I think, a voltage dye imaging. So these are both imaging experiments. And you're looking at activity when you apply a particular odorant. Okay, so when you apply a particular odor, you get a response in a particular glomerulus, kind of what you would expect based on that convergence. But then if you apply a different odor, you get a different pattern. But if you apply another odor, you get another pattern, but there's some overlap, right? So you get activity here for odor B, and some activity here for odor C. And it's not just one glomerulus. Now you've kind of got two glomeruli. So it's not as, you know, one-to-one thing as you might expect from the anatomy. And then here, the similar property. So it's C3, 4, 5, 6. This is using odorants, chemicals of increasing carbon length. So this is 3 carbons, 4 carbons, and so on. I can't remember if this is an aldehyde or an alcohol, but you have those linear carbon chains that you can then lengthen. So people like to use these because it gives you kind of that illusion of linearity, linear dimension, but it's not that simple. Okay, so C3, you get a response kind of here. Each of these sort of dark spots is activity in the glomeruli, here and here. It's kind of summarized on the right here, color-coded. And so basically you can see that you have changes in activity patterns depending on the odorants, but it's not quite as simple as a one-to-one effect. Okay, so what's going on here? So can we call olfactory processing? There's this term that's used frequently as a labeled line. Okay, so labeled line is the idea that you have a particular stimulus that activates a particular protein, that activates a particular olfactory receptor neuron, that activates a particular glomerulus, that activates a particular mitral cell, and so, you know, boom, you're done. And there are some sensory systems where that seems to be the case, and olfaction really doesn't seem to work quite like that. Okay? So if you remember each olfactory receptor protein, remember this is a chemical binding reaction. What do we know about chemical binding? It's not a perfect locking key. That the key that's sort of a little similar to the perfect key might unlock a little bit, right? All of the receptors kind of work that way. You have something that's close, you're going to get some activation. So you have maybe a best olfactory, a best odorant, but you might have a number of other odorants that are kind of similar that might get some activation. So olfactory receptor protein might bind multiple things to different degrees. And so you really kind of reduced that sort of simple story. So now olfactory receptor neurons are going to respond to a few different things and so on. And in a lot of systems, the equivalent mitral cell actually gets multiple input from different glomeruli, so it's not quite the same. Okay. And everything gets worse with intensity. So the larger, the higher the concentration, the less specific the binding process is. Okay, so let's look at an example of this. So this is all amyl acetate being provided to the system in increasing concentrations. And so you can see that the what used to be a very specific response, this little spot up here, becomes 2 spots, larger spots, little activity, and so on. And at some point, you're getting a very, a large response pattern. And then, of course, if you do this with 2 different odors, then you have enormous overlap in the response patterns. Okay, so it looks like what you have is you have this sort of very broad binding at the protein level, broad coding at the receptor level, and so you don't have that label line mapping. And so this has often been referred to that you have some sort of combinatorial code, that it's the particular pattern of responses that are responsible for detecting a particular odor. And of course, this all gets a little messier when you start talking about blends, right? So if this is all we're talking about single chemicals. You start talking about blends, it starts to get kind of muddy. And so how are you gonna deal with this? How does it even make sense to analyze in the sense of specific chemical compounds? Okay, so we're going to talk about how you might sort of solve this problem using what I'm going to call sparse coding. Okay, so the idea is that at the lower levels you have very distributed and broadly tuned coding, but at a second level, at the second relay, we're going to use a different style of coding that we're going to call sparse coding. And we're really not going to talk about this, but there's a lot of evidence that the way that you get to sparse coding from distributed coding is to use neural synchronization, so the timing as a parameter for coding in the system. Okay. And then you can use this sparse representation in order to do complex pattern analysis, right? And this is something that the theory goes that this is what you use for memory and recognition, that you have a sparse network, and you can put a lot of information into a sparse network, whereas with a very densely coded network, you have too much overlap and you can't tell the difference between different stimuli. Okay, so let me sort of briefly give you a little whirlwind tour of one of the systems where this has been sort of studied intensely, and this is the insect olfactory system. So I just first want to say that there's a lot of anatomical similarities between the mammalian system and the insect system, even though insects are really quite different from mammals. They don't have noses, they have antennae. So sort of the external structures are quite different. But the pattern in the anatomical structures and the way that things are being encoded is quite similar. So this tells you a little something about the evolution of this sense, right? That these animals have to solve the same problem. And they've kind of come up with similar solutions in order to do it. Okay, so in the insect system, again, you have olfactory receptor neurons that are out in the antennae. Okay, and those project to the equivalent of the olfactory bulb, which is called the antennal lobe in the insect, and that's this structure down here. Okay, so it's actually this neuropil area, and again you have the glomerular system that's not really shown in this representation. So olfactory receptor neurons contact the projection neurons, these PNs, projection neurons, the main principal neurons in the antennal lobe, and those project onto the next area, which in insects is known as the mushroom body. And we haven't quite gotten to this in the mammalian system. We'll come back to this. And the idea is that those projection neurons have a very sparse synaptic connectivity with an enormous number of neurons in the mushroom body. So there's something like let's see if I remember, like like 700 to 50,000 neurons. So there's an enormous amount of divergence there. And they're sparsely connected. Okay, so every principal neuron in the mushroom body, receives input from a set of projection neurons. But projection neurons only project to a small subset of the principal neurons in the mushroom body. Okay. And so if you record activity in the antennal lobe, kind of remarkably, remember the olfactory receptor neurons, you have this sort of slow response, it's depolarizing, it fires excitedly, and then it stops. When you get to the antennal lobe, you suddenly have this profusion of temporal patterns. So these are spike rasters, okay? And so each of these groups of rasters is a response by one neuron to a range of different odors, okay? And the odor, if you can, I don't know if you can see that, there's sort of a gray bar right here. It sort of goes from here to here. Okay, and that's the stimulus presentation. And then you're recording over a much longer time period. Okay, and what's the scale here? The scale is something like half a second or something. Okay, and so this is one neuron responding to a whole pattern of odorants. And you can see that there's a real diversity in responses. Sometimes it'll basically kind of shut off to a particular odorant. Sometimes it'll fire really actively. Sometimes it'll fire for a long time. Sometimes you'll get very strong firing and then inhibition afterwards. And so you can really tell which odorant it was based on the pattern of the response. But it's responding in some way to lots of different odors. Yes?
Yeah, so what's happening along the y-axis in this graph?
Oh, sorry. So this is a raster plot. So each of these little tick marks is an action potential. So you determine the time of the action potential. Each line of ticks is a trial. Okay, so this is something like 10 trials for this particular odor. And then this is another 10 trials for a different odor, and another 10 trials, and so on. Does that make sense? Okay, and then so you have time on this axis and trial number on this. Okay, so you can see this is very broadly tuned. There's temporal patterns. But if you record at the next level, that's getting direct input from these neurons. Okay, so this is one of those mushroom body neurons. What you can find is that it responds only to one or two of those odors, and it responds in a very sparse way, a couple of spikes. It's reliable, right? So it responds to each of those stimuli, but it's very sparse. So when I say sparse, this is what I mean. It's a very sparse response. There's less firing, there's greater specificity, and as a result, if you record from multiple neurons, there's less overlap in their tuning properties. Right? They're looking more and more unique. Okay, so just to go with I showed this slide before, but this is just what we mean by this sparse coding is useful discriminating large numbers of stimuli. So now if you look at the second level, you have a pretty good idea of what the stimulus was. All right, so kind of returning back to the mammalian system from here. Okay, what happens after the olfactory bulb? So the olfactory bulb, like in the insect system, you have a projection from the olfactory bulb to what we call the piriform cortex. Sometimes this is spelled with a Y, sometimes it's an I, it doesn't really matter. Receive direct input from the olfactory bulb via the olfactory tract. And this is sort of thought of as the primary olfactory cortex. And here, so that beautiful mapping that we saw in the olfactory bulb is completely gone. I'll show you a picture of this. And so it turns out you didn't really need that mapping, but again, you have this sort of sparse coding in the piriform cortex. So very similar to what I just showed you in the mushroom lab. From there, you also have entorhinal cortex, also gets some direct input from olfactory bulb that goes on to the hippocampal formation. And so we'll learn later, hippocampus is a site for memory formation or memory creation. And so really your nose is just basically 3 synapses from this memory center. So this might account for the fact that odorants are very salient in evoking particular memory senses. Right? So it's sort of the Proust effect. Olfactory bulb also can project to the amygdala, so you can have very direct connection to sort of an emotional response to different odors. Is there a question? Yes?
Why does it, like, I'm confused, like, why does the paraform just kind of throw that information away? Because it seems like it would be, like, kind of important, like, or does it just, like, what's, like, the evolutionary significance of just, like, mapping it out and then being like, I don't mind them?
So the question is, what's the evolutionary significance of creating those beautiful maps and then throwing it away? Let's hold on to that question for a minute. Let me show you the data. Okay, so here's the anatomy of this projection. Here's your olfactory bulb. If you remember your anatomy from Unit 1, that's that bulb is sort of sitting there on the ventral surface of the brain. And it sends, this is known as the olfactory nerve, but it's not really a nerve, it's sort of like this big thick tract, olfactory tract, that goes to the piriform cortex, which is sort of located under the temporal lobe right here, okay, amygdala, and then finally entorhinal cortex. Here's kind of a block diagram of all of its projections. So here's piriform cortex with a Y, olfactory tubercle, I don't remember what this does, amygdala, entorhinal cortex, sends information to hippocampal formation, and so on. There's also a projection from these to thalamus, which then sends a projection up to the cortical areas. There's also an anterior olfactory nucleus. Okay, so let's look at piriform cortex, so basic olfactory cortex. LOT stands for the lateral olfactory tract. So there's basically a big ipsilateral projection to the piriform cortex, pretty much to the anterior part of the piriform cortex. And the piriform cortex is kind of a funny place. It's, remember, mostly what we talked about so far is neocortex. It's that 6-layered cortex. Piriform cortex only has 3 layers. And so piriform, along with several other things, are considered archicortex. Okay, so archi meaning first? Early cortex. So this is old cortex. Okay, along with the hippocampus, which also doesn't have 6 layers. So it's a 3-layered structure. Okay, and unusually, we talked about the neocortex, where layer 4 was really the input layer. Well, in this case, the LOT travels along the first layer, and so it makes projections onto the principal cells in this top layer, layer 1A. And here's your principal neurons of the cortex. These are the pyramidal neurons. Okay. And then there are some deep pyramidal neurons. So this is in layer 2, some more neurons in layer 3, and then those project outward. Okay, so layer 2 really contains those neurons that receive inputs. And then within the piriform cortex, you have a lot of collaterals. So there's a lot of sort of collateral associated fibers. So it's, in that sense, it's very similar to the mushroom body that we showed you before, where you have this very distributed projections from the olfactory bulb, and then there's actually a lot of interconnections as well, and especially from anterior to posterior that receives input. And so the idea is that you have these collaterals that form basically like a network. So I'm sure all of you have heard of neural networks. It's, oftentimes it's shown sort of an all-to-all network, or an all to sparsely to lots of others, sort of randomly connected. And this is really kind of an incantation of that, incarnation of that you have this sort of sparse network. And it's thought of as an association network. Okay, so here's kind of a cartoon of the loss of that mapping. Olfactory epithelium, interspersed olfactory receptor neurons that then project in a very precise map. And then these project to the piriform cortex, and you've kind of lost the mapping again. This is some calcium imaging. So here's a sort of a surface image of piriform cortex. This is the response of these neurons in response to 3 different odorants, and you can see that they're quite interspersed, sparse and interspersed. So again, you have an association cortex that has a very sparse representation. And so it's kind of getting back to the question of, well, why would you create this map just in order to kind of throw it away? I don't have a really good answer for that. In the sort of the second half of this talk, we're going to talk a little bit about coding. So what good is a map? Why would you have any maps at all in your brain? Yes?
You can create more diverse patterns.
You can create more diverse patterns. Okay. All right. What do you think?
Compartmentalization.
Compartmentalization. Okay. Why is it good to compartmentalize?
Oh, okay. Because like you're more efficient that way. You're able your functions won't overlap per se. So if like you have like a set of odors that respond to mating, right? That won't overlap with like say a set of odors that respond to food, right? You can have all the mating odors go to the mating place, and then they'll respond with mating behavior, and you can have a very efficient and very rapid response.
Okay. So efficiency. There's an argument for efficiency. There's an argument for not mixing up your signals. We had a what we.
You were just gonna say what I was gonna say, but I do have another question. So, like when you're saying that it has to do with mapping in the cortex, is there like a temporal component too? So do different smells produce like a different pattern of depolarization that actually affect it differently?
Yeah. So the question is, what about temporal patterning of the activity? Yeah, so these are all really good points, and I don't have a good answer for why you would create a wonderful map and then destroy the wonderful map. But I think part of the answer is that yes, you can also use the temporal dimension to do coding, and I think the best argument for that is in the olfactory system. But yeah, actually it's kind of an open question, like when would you get good maps? And I think a lot of it has to do with the efficiency in the sense of a wiring diagram, right? So you could create any kind of network you want based on simply connections. So any 2 neurons could connect to any 2 other neurons, but you may want to conserve on how much wiring you're using to do that. And so in this system, it might make a lot of sense to bring all of the neurons together to minimize the wiring, to do lateral interactions. And in this area, it makes sense to have a very widely sparse network because of what you're trying to do at each of these levels. So it kind of speaks to what kind of processing is happening at each of these levels.
Can the piriform store memory?
Can the piriform store memory? I don't know. We can maybe come back to questions like that when we talk about hippocampus and memory, because the sort of for a while the going theory was that all of your memory was in the hippocampus.
Yeah.
And now the idea is more that your hippocampus is not storing memories per se, but consolidating memories and having contact with neocortex, where the memories are stored. And so piriform cortex might function the same way. Yeah, there's a question.
Yeah, could we explain what you mean by lateral interactions?
So lateral interactions, and we're not going to talk about this too much today, but it's actually really well known. I probably should put this into future lectures. So you have mitral cells, and that send their axons out, and they have 2 types of branching. So they have an apical dendrite that's located in the glomerulus. Okay, that's receiving input from the olfactory receptor neuron. And you have another glomerulus another mitral cell. And some lateral interactions. There's sort of two types of lateral interactions. You have periglomerular cells that contact here and here. And so there are reciprocal interactions in the neuropil. It's somewhat complicated, but there's reciprocal interactions. So if you get activity here, the periglomerular cell can inhibit another glomerulus. Okay, so it usually contacts multiple glomeruli. And then you have your granule cells. And both of these are GABAergic. Okay, and then you have granule cells, and here you have sort of what's sort of classically known now as a dendrodendritic synapse. So you have reciprocal synapses between the basal dendrites of the mitral cell and the dendrites of the granule cell. So this is excitatory, this is inhibitory, but you can also get lateral effects there. So there's sort of two ways to get lateral effects. And there's sort of two schools of thought as to what these lateral interactions are doing. One is that you're sharpening the tuning. So say you had, a C3 carbon length response here and a C4 here, that you might be able to, have a winner take all. So you can identify C3 and suppress responses to C4. That's one theory. But we know most of the chemicals kind of don't work like that. And so the other theory is that these are involved in temporal responses. We know that there are all kinds of oscillations in the olfactory system. And we know that there is neural synchronization. So the granule cells may be acting to synchronize responses across different mitral cells, or synchronize mitral to granule cells. So you have something that's excitatory neuron that contacts an inhibitory neuron, and we know that you can get oscillations out of a system like that. But people are still kind of arguing over what's happening there. This also provides a site for top-down control of the olfactory bulb. So there's a lot of descending efferents that go back to the olfactory bulb. And so there's a lot of modulation that can happen there. Okay, so this is basically the end of olfaction. Are there any other questions? There's a lot we don't know. And it's a really interesting area. But let's move on to taste. Okay, so taste is a completely separate sense. So most of what we kind of think of as taste is really a mixture of taste and olfaction. So the flavor of foods and so on. Okay. Okay, and this is something that I'm sure you're familiar with from all the way back to grade school. But you have these 5 different gustatory senses: salt, sweet, sour, bitter, and umami. So is umami is everybody comfortable with that? It's like in my lifetime this was kind of a new thing, but you guys have probably learned this from grade school. Is this a grade school thing now? You learned umami? Okay. Great, because I like it. Okay, so, and umami is kind of funny, you can tell from the name it was sort of discovered by the Japanese. And it's a sort of savory sense, and it's really a detection of amino acids. So it's part of that sort of meaty sense that you get from meat and mushrooms and things like that. Okay, and then bitter is another sort of complex. These are all quite simple, but these are sort of a couple of complex senses. And so things like quinine, if you like your gin and tonics, strychnine, but it's generally there to detect things that you don't want to eat. So it's very sort of a protective sense. There's lots of poisonous alkaloids contained in plants. So if you sort of go around tasting lots of plants, you want to avoid things that taste bitter. Of course, now we all sort of have developed a taste for things that are bitter, like coffee or cilantro or things like that. All right, and so this is sort of your classic representation that different aspects of taste are at different parts of your tongue. I don't think this is really all that an important thing, but it's a thing that you see in every textbook. So there it is. Okay, and part of the idea is that taste basically can come down to what's known as the hedonic value. Is it good? Is it bad? Do you like it? Do you dislike it? And this is important because you have to know, is the thing that I'm eating going to kill me? Or should I spit it out right now? Okay, so everybody knows what a taste bud is, except you don't. What we think of as a taste bud is actually a papilla, so this sort of, anatomical structure. The taste bud is actually this little conglomeration of taste cells within the papilla. Okay, and so you have this collection of taste cells that receive input through the taste pore, and unlike the olfactory neurons, they actually make synapses. So the taste cells themselves don't have axons; they have synaptic contacts onto gustatory afferents. Okay, and so they have several different transductions. So the different types of taste really reflect different transduction mechanisms. So I want to take a moment here to emphasize that even though this diagram is showing all of these things in one taste cell, these are actually segregated into different taste cells. So don't be, yes?
How many taste cells per papilla are we talking about?
Oh, I don't know. I don't know.
How many different? It's like no, not quantity. How many different types?
Oh, well, it would basically come down to those 5 main types. Yeah.
So it's isolated like one papilla. What would we expect this to be?
So a papilla, I don't actually know. I think a papilla might have different types of taste buds. I actually don't know. I'll have to look that up. Yeah. So each taste cell is going to detect one type of chemical response, one type of transduction. But I actually don't know if they're intermixed. Sorry.
You learn about taste as mapping in the tongue.
So the taste, yeah, if you go back to that tongue issue, right? So you're going to have a higher density, but they're not completely separate here. So I actually don't know the answer to that question. Let me see. The question is, if I'm getting it correct, is taste segregated by papilla? Yeah, I actually don't know that. Does anybody know? All right. You can check the textbook. Probably says somewhere in the textbook. Yeah, so there are different types of papilla. I wasn't gonna talk about that. Let's talk about transduction. Okay, so there are different types of transduction. So each of these types of taste really depend on different transduction mechanisms, but we can kind of classify them into 2 sort of types. The salt and acid taste, salt and sour, use ionotropic channels, and these really sort of are about as simple as you can come up with. They directly pass electrons or protons in order to depolarize. And we'll look at this closely in a minute. And then the other type uses different variations on 7 transmembrane receptors that are linked to G proteins. That's the sweet, bitter, and umami. Okay, so you have the initial transduction occurs sort of out here in this apical domain, and then you have the rest of the receptor, kind of similar to what we've seen in hair cells and in photoreceptors. You have this sort of polarization, division of labor in the different for the transduction mechanisms. Okay, and we'll look at this more closely in a minute. Like I said, this diagram's a little misleading because you actually have each of these transduction mechanisms separated into different taste cells. Okay, and so at the bottom end, you have the end result is depolarization through one means or another, and neurotransmitter release. Kind of unusually, the neurotransmitters doesn't seem to be very well known, but it uses things like serotonin, ATP, or GABA in order to transmit information to the afferents. Okay, a little unusual, so it shows serotonin receptor here. And then there are 3 cranial nerves that convey this information. Cranial nerve 7, 9, and 10. Okay, so let's look at each of these in turn. So salt, so the taste of salt is basically conveyed by a sodium channel. This is a particular kind of sodium channel. It's called amiloride sensitive sodium channel. It's a 2-membrane, 2 transmembrane domain receptor. It's often found in epithelial cells, so it's involved in regulation of salt and water in epithelial cells, but it's found in these particular taste cells. So when you have high degrees of salt, you can imagine that they cross the membrane and cause depolarization. It's about as straightforward a mechanism as you could imagine. Sour, you all know that acids have high proton counts. And so when you have a lot of protons in the extracellular membrane, it passes through this channel. This happens to be a TRP channel, okay, a proton-sensitive channel. And again, you have depolarization of the cell. So very, very straightforward. Okay, so sweet is conveyed by, sweet and amino acids are conveyed by a set of these families called the T1R family. Okay, and there's variation in the subunits, so there's some overlap here, but there's variation in particular subunits that are involved that discriminate between different types. So the sweet glucose is detected by a combination of the T1R2 and T1R3. So it's a heterodimer here. Whereas amino acids use the T1R1 and T1R3. So a slight different version of the same dimer. In both cases, you have a G protein. The G protein activates phospholipase C that generates an IP3 second messenger that then opens a particular TRP channel, in this case TRPN5. Okay. Here you have calcium influx, and that causes depolarization. Yes?
So like, with like odors and stuff, there's like very like characteristic mapping, right? And all the neurons pretty much have like the same mechanism, but with the way they were mapped, that's how you got the precepts. Can you explain like how you get the precept of sweet and bitter and stuff like that? Because they all just seem to just kind of blend in with the stream of cranial nerves
Yeah.
going back to the brain.
Yeah, no. The question is, can you explain how you get the percept of sweet or bitter? Not really. It's really not a very well-known system.
Okay.
We'll look at some of the projections. I mean, obviously, you can tell the difference between something that's sweet and something that's salty.
Yeah.
So, but oftentimes you can't really tell the difference between different sweet things, right? A lot of what we perceive as different types of sweetness really has an olfactory component.
Okay.
So it's really kind of a coarse sense in that way.
So if I like shut off like your nose, can you not perceive certain tastes as well?
Yeah, how many people have done the apple onion thing? Yeah, but how many people know what I'm talking about with the apple onion thing? Oh, really? Okay. Okay, your homework, okay, is to go home and have your roommate hand you either an apple or an onion, raw, and you're supposed to hold your nose and you take a bite and see how long it takes you to figure out that you're biting into an apple versus an onion. I want to report back to see how you do. No, I mean it's actually quite evocative. I haven't done this myself because I'm not into self-experimentation.
So, along the same line, how do artificial substances that have like a completely different chemical structure from, say, like sucrose?
Yeah, I don't actually know. Yeah. So sucrose can often be pretty quickly broken down into glucose. So I suspect that this is primarily a glucose.
But if you taste glucose, it doesn't taste good.
Yeah, there may be a concentration effect there. So for a lot of olfactory percepts and sweet and taste percepts, if it's too strong, it's perceived as bad because you're basically overstimulating the system.
So do you think that in the mouth it might rapidly
I suspect
metabolize
so,
into
yeah.
glucose? But wouldn't that not make it an artificial sweetener, and wouldn't it have a caloric effect too? If it's due to absorbing glucose instead of an artificial
You know,
sweetener?
I'm not really sure. Maybe it is sucrose. Yeah, yeah. I don't know exactly. Maybe you're right. Maybe it is a sucrose and the glucose is. I'll have to look it up. Yeah. Yeah.
Since these two senses have the same channel opening at the end, how can your body tell which one?
Yeah. So this is where I think segregated expression of these two types of receptors, and there's evidence that you either express this dimer for amino acid detection or this dimer for sweet detection, and not both in the same taste cell. So that's where the segregation to particular taste cells is important. So somehow this has to be regulated, right? Because otherwise, obviously, you wouldn't be able to tell the difference which receptor you would be stimulating because they converge onto the same second messenger pathway. Yeah. Another question? Yeah, okay. Okay, and just to complicate matters, if you have the T2R receptor, so remember the last one was the T1R. This is a T2R that detects bitter compounds, and again, it has the same sort of second messenger pathway, BSCI353 trig channels. So it's pretty clear that in order to segregate those, to detect one versus another, that you have to have segregation of the transduction mechanisms into different taste cells. And then you have that sort of, this may actually be sort of a labeled line system where the bitter taste cell synapses onto an afferent that then carries bitter information into the central system. All right. So the central pathway, so we mentioned that there are these three cranial nerves that carry different types, carry information. These are related to the different papillae. Okay. And those project to the brainstem. So there's a nucleus of the solitary tract, or more simply a gustatory nucleus. So you have this brainstem nucleus that receives information, that sends it on to the thalamus. And the thalamus sends this information on to the cortex, so projects to this insular cortex, where taste information is processed. Okay, but there are other projections from the brainstem nuclei to things like the hypothalamus, which is involved in appetitive behavior. Okay, and the amygdala, where it can affect the sort of affective behaviors. So you can loop in those aspects of eating and your hedonic sense and so on. Okay, and then sort of one last aspect of chemical senses is the idea that you there's a whole nociceptive system for things that are irritants. Okay, so we all know that if you get ammonia up your nose, it doesn't. You can detect it, you can smell it, but it's mostly you get irritation, right? It's bad. And so you have these free nerve endings in your nose and in your palate that are sensitive to various kinds of pollutants. We mentioned earlier capsaicin, one of those trip channels that can detect capsaicin, so you get that sort of hot sensation. And this is all conveyed via the trigeminal nerve. And so this is thought of as sort of the trigeminal system for nociceptive chemicals. Okay, so just to kind of wrap up, so when we think of as taste, it's really sort of a complex taste olfactory system or flavor. And one sort of last note is that there can be a clinical loss of smell. We call this anosmia. And one kind of interesting thing in recent years, people have started to make connections between the loss of the sense of smell, especially in older people. As you get older, you're more likely to lose your sense of smell. And that this might be actually an early indicator of neurodegeneration. There's some thought that it's linked possibly to Parkinson's and so on. And so if you lose your sense of smell, this is something that your doctor kind of wants to know. And kind of part of the theory is that this might be related to the idea that you have regeneration in the system. It's very exposed to the environment, and you need regeneration, and that if you are no longer able to do those sorts of regeneration, that is an indicator that something's kind of gone wrong in the system. All right, so, oh my goodness, okay. So we have gone through olfaction.